Just wanted to see how many N's I could get into the title. Yeah, so thank you very much for kind of inviting me along. Before I get into the kind of details of the talk, I'm going to work out how to change slides. There you go. I'll just give you a very kind of quick overview of what I'm interested in research in general, and then you'll see how this talk kind of fits into that more specifically. So I'm very motivated by kind of image analysis, and in particular at the moment, multiplex images, so like this one up on the left. So kind of looking at where different cells are and what they are, but then also using mathematical modeling to kind of try and understand the processes behind spatial localization of cells. And then at the same time, taking kind of statistical approaches to describe spatial localization, which can then be applied to both imaging data or to modeling data. So just as a kind of motivation of why I think spatial analyses are important. So I absolutely love this, this kind of GIF, because every single frame of this has exactly the same mean X coordinate, mean Y coordinate, standard deviations, in X and Y directions, same correlation coefficient. But obviously the patterning that's going on there is very different. So if you're not taking into account kind of spatial effects when you look at statistical descriptions of data, you could well be missing the kind of dinosaur that's hiding under the surface. And in terms of kind of how this fits with medical imaging, well, we've got this incredibly high spatial resolution in histological data, and now in kind of multiplex histology and kind of other multiplex techniques. But we're not using a lot of that information. And with kind of advances in image analysis, we can now pull out XY coordinates of all of the cells, but we're maybe not using that. And if we can kind of automate that type of description, then we can do more sophisticated analyses. And you know, in an ideal world, things which are reproducible and quantitative. So that's kind of where my research lives in general. And I tend to think of what I'm trying to do as kind of fitting into this cycle where we've got the kind of data environments on the left, where we want a workflow of spatial analysis tools that we can apply to data, ideally in the clinic or in other research settings. But also on the right, we've got these kind of mathematical and computational environments. And what I'm really trying to do is use mathematical models, so particularly agent-based models, to kind of generate synthetic data where I can control what's going on and understand what's there, and then test new statistical techniques on that data. Which brings us to this talk. So you're going to get a little bit of biology, a little bit of modeling, and a little bit of statistics. So a little bit of all three of these things. And this is the last time I'll show the dinosaur, sorry. So the motivation for this, so some of my collaborators have got a laser, which I find incredibly cool and is kind of very anticlimactic maybe, but they're doing something called imaging mass cytometry, which is a great way of getting multiplex data. And it basically works by putting, instead of kind of traditional attaching a big blob of dye to a cell and looking down a microscope, you're attaching kind of heavy metal, you know, single ions or single atoms or whatever it may be, and then you blast them with a laser. And the stuff that comes off, you put through mass cytometry, and that gives you, at a kind of 1 micron scale, a kind of picture of what's going on within the sample. But you don't just get, as you see here, kind of red, green, blue. You get 40, 50 different color channels associated with this. So mathematically, you can think of every point in the image as being associated with some big vector. And that might include kind of discrete markers. So you might look at it as a pathologist and say, oh yes, this combination says this cell is a blood vessel or not. Or it might be a continuous marker. So, oh, I can see that this cell is expressing CD3, and there's more or less, but it's living on some scale. So this is the kind of mathematical object that I'm very interested in, is if I've got high dimensional labels, which may be discrete or may be continuous, how can I go about kind of describing what's going on with that? So in terms of this picture, we're now living firmly in the bottom right corner. So I'm not going to talk to you much about any real data here. I'm going to talk to you about a kind of model simulated system that we've developed to kind of generate some data of this type. And then I'm really interested in looking at, well, if I have this incredibly high resolution multiplex data, what can I get out of that? Versus if I had kind of standard IHC or something along those lines. So in particular, I want to model some kind of interesting system, and I want something where spatial localization is important and where some sort of continuous phenotype variable is important. And I want it to involve macrophages so that I can have lots of M's in my title. So in particular, we've kind of focused in on this paper all about kind of how macrophages help aid intravasation of tumor cells. So this is the kind of graphical abstract from this paper. Essentially, what happens is at the start, day 0, we see macrophages leaving blood vessels, and they come out with what's kind of historically known as a sort of M1 phenotype. So an anti-tumor phenotype. They're responding to CSF1 or CCL2 or things that are kind of produced by the tumor. And that macrophage then wants to go and invade and attack and be generally anti-tumor. And what happens is, while it's inside the kind of tumor mass, it's being exposed to things like TGF-beta, which are changing the phenotype of the macrophage. And historically, you'd say, is it M1? Is it M2? Well, now it's an M2 macrophage. That means kind of pro-tumor. It's not attacking anymore. And in fact, what they do in this system is they move their way back up towards blood vessels, or right, towards fibroblasts associated with blood vessels. And once they're hanging out in the blood vessels, they're also attracting tumor cells. So you end up with tumor cells following these M2 macrophages back towards the blood vessels. At which point the cancer cells can kind of go into the vasculature and make their way around to try and metastasize. So this is a really interesting system because there's a lot of kind of different spatial elements involved. We're interested in, is the macrophage next to a blood vessel? Well, that may mean that it's a kind of very anti-tumor macrophage, which is just extravasated. Or it may have been hanging around for a week or 2 weeks. It's gone into the tumor, it's changed phenotype, it's come back out again. And obviously, if we're just looking at traditional histochemistry that says, is it a macrophage or not? We can't necessarily tell that. We just see a big brown blob. If we're looking at multiplex data, maybe we can really infer exactly kind of what's going on within a macrophage and kind of assign some more continuous label to it. So from a modeling point of view, that's exactly what I'm going to do. So we've got a model here of cancer cells, which are kind of growing based on a kind of oxygen-based cell cycle. And they're just kind of embedded in this world of stromal cells that really are just kind of providing, I should stop hitting that, just providing some resistance to them. And then you'll also see some necrotic cells. So when a cell dies, it's going to occupy space for a bit of time. And our kind of really interesting ones here are the macrophages. So rather than just saying M1, M2, all of these macrophages have a kind of phenotype going on within the macrophage, which is describing how it's going to behave and kind of determining that. And I'm also thinking of just some fixed coordinates, which are blood vessels, where the macrophages are going to kind of enter the simulation from. Oh, and there's an equation in the top because I'm solving some forces here. And I thought I should have a slide full of equations because this is maths talk, but really these are the kind of diffusible species that are going on underneath. So we've got CSF1, which is recruiting macrophages and kind of attracting them into the tumor. We have TGF-beta, which is changing macrophage phenotype. And then we have EGF, which is what's kind of recruiting the tumor cells towards the M2 macrophages. And finally, CXCL12, which is bringing them back out. You don't need to remember all of these. In terms of this phenotype variable, so I'm just saying all of these subcellular processes, which are describing macrophage phenotype, I'm just going to bundle into one variable P and live somewhere between 0 and 1. So at the P equals 0 end of this spectrum, I'm thinking of M1 macrophages. So they're going to do a lot of cell killing. They're not really attracting tumor cells towards them. They're being very attracted to the tumor, and they're not coming back towards the vessels. Whereas at the other end of the spectrum, it's the exact opposite. And then there are some more forces. So this is just, if you're interested in exactly how I've kind of implemented this, I can talk to you about that in a bit more detail later, I guess. So the key picture is what's going on in the top right here. Macrophages coming out, going into the tumor as a yellow anti-tumor macrophage. They kind of get converted across the spectrum, and then they come back out to the blood vessels, bringing tumor cells with them. So in terms of what this looks like, well, this is a typical kind of realization. So for early stages, we have the tumor kind of happily growing away, and we have these pink crosses of blood vessels around the edge. You can see as the kind of CSF concentration goes up, we start to get these little yellow macrophages appearing. And then eventually they start to get recruited into that tumor mass up a kind of chemotactic gradient. Once they're in there, they start attacking. And you can see they kind of change along the spectrum, going from yellow towards blue. And the blue ones are going to come back out again. And you can see as they do, they kind of change the shape of this tumor. So these red tumor cells are following them back out towards blood vessels, while being, in this case, kind of eaten by a lot of macrophages. Yeah. So what I'm very interested in is kind of what's going on at the edges of these. So we talked to one of our collaborators, Parameter Sweep, who is very important in this work. So what we're doing here is we're just changing these two parameters. I'm very proud of Parameter Sweep, by the way. I think that's a good joke. So all that I'm doing in this parameter sweep, I'm not changing any parameters to do with the tumor growth. So the underlying tumor model is exactly the same. The only thing I'm changing is how much are my macrophages attracted to tumor cells, or how heavily are they being recruited? And you can see kind of in this, along this kind of bottom axis, if I kind of don't have very strong attraction to the tumor, obviously the macrophages aren't going there. It just kind of grows up into a kind of compact mass by itself. Whereas as I kind of increase the recruitment to the tumor, all of these anti-tumor macrophages are getting in and wiping it out. In this kind of top right corner of this space is where we get the really interesting stuff happening, where we get this kind of very diffuse shapes, and we get these kind of interesting perivascular niches. So this is just a kind of magnified version of one of these. And what I'm really interested in are, when are the simulations where we're getting this kind of behavior, where my tumor cells, my M2 macrophages, and my blood vessels are all kind of in close proximity. Because really those are the kind of simulations where we're seeing this kind of potentially metastatic behavior, whereas in the bottom row here, perhaps that's less of an effect. So really this is my kind of synthetic data. I want to try and look at how I can characterize this. So if I just look at cell counts, well, the pink bars that you see here are number of tumor cells, and the brown bars are number of macrophages, and that's blinded to phenotype. And you can see, we can pick up when there's tumor elimination because there's no tumor cells, but we can't really tell the difference between the red box in the top right, the kind of diffuse tumors, and the simulations in the bottom where actually we've just got this kind of compact mass. So we definitely need something more refined than just cell counting. Similarly, if we look at the kind of distribution of macrophage phenotype, so this is a bit of a proxy of what you might be able to get from doing kind of looking at RNA in single cells, but in a non-spatial way. If you can understand how these macrophages are behaving, but not see where they are. And you get more of a sense of, well, in the bottom here, we've just got M1 macrophages, we've just got the anti-tumor stuff going on. Whereas in these higher simulations, we're seeing more of a split between pro and anti-tumor. But again, we can't really distinguish between the blue and the red boxes, the tumor elimination and the kind of diffuse growth, just from these statistics alone. So the question then is, well, those are not taking into account space. So let's try using some spatial statistics to describe these things. So in particular, I'm going to use this cross-pair correlation function, which I think is a great tool for kind of telling you essentially where two different types of cell are relative to one another. So essentially the way this works, if you have, say, blue cells and red cells, well, you think about if I sit on top of a blue cell and I look some distance r away, how many red cells do I see? And in particular, I'm interested in how many red cells do I see, but compared to some kind of null distribution. So if all of the red cells are just spread around randomly, am I seeing more or less than I would expect? And that's essentially what this PCF plots. So values above 1 are saying I'm seeing more red cells at distance r than I would anticipate seeing. Values below 1 are I'm seeing less. So we kind of have a sense of clustering or exclusion at a different length scale. And mathematically, how this works, well, we're basically just counting the number of cells that are within our kind of annulus, and then we're averaging that over all of our other cell types and comparing it to spatial randomness. So I'm going to come back to this equation in a minute. So what this looks like, so this is actually from some real data. You can really pull out, okay, are my two cell types clustering together at whatever length scale, in which case they're going to be above 1, or excluding one another, in which case they'll be below 1. So we can apply this to our synthetic data. So the PCFs you're seeing here are from tumor cells to blood vessels. So these are just discrete cell types. And we can really kind of start to pull out, well, actually in the bottom row here, we're seeing 0 for quite a long range of kind of length scales. And then it kind of peaks up at a distance. And that's really saying that in these bottom simulations, the tumor cells and the blood vessels are staying a long way apart. Whereas in our kind of red box up in the top, we see a spike at a very short distance. So that's telling us if I look a very short distance away from a blood vessel, I'm quite likely to see a tumor cell. So we're kind of starting to pull out this kind of perivascular niche a bit more in terms of tumor cells and blood vessels.
This is the final time point.
Yeah, so this is all from one time point. Yeah.
But it's far into the simulation, it's not.
Yes, yeah. So this is all specifically these time points. Yeah. So we can do this as a time series.
You just wanted to bring it back to this. I know.
City and Sweep are coming back, don't worry. Yeah. Yeah, so the problem we've got with just using PCFs to do this is if I want to look at now macrophages to blood vessels, well, in this case, I don't have a concept of macrophage phenotype. I just know macrophage, don't know how it's behaving. And that's something that the PCF can deal with because it's got two different cell populations, macrophage tumor cell, or sorry, macrophage blood vessel in this case. But you can see it's not especially informative. Well, if you're familiar with PCFs, you can see it's not very informative. You know, a lot of these things look very similar. And really the problem is that you're not able to distinguish between M1 macrophages, which have just kind of left the simulation, and M2 macrophages, which have really moved back into this perivascular niche. So the problem is we really can't split this up very efficiently. So if we, this is the kind of thing that you might be able to calculate from just an IHC slide. So if I have macrophage locations and blood vessel locations, so I just need two markers, I could calculate this, but it's not massively informative. Whereas if I have this multiplex histology and I really understand what type of macrophage I'm looking at, I want to do something that's kind of taking that information into account. Now, historically, what you might do is just say, well, okay, let's have two populations of macrophage, M1, M2, and split them up like that. The trouble is, the more detail you want to look at, the more populations you have to split your macrophages into. So if I want to say, you know, macrophages with P equals 0, P equals 0.5, and P equals 1, I need 3 populations. And you very quickly run into problems because these aren't discrete populations. They're one population described with a continuous label. So this is where the kind of interesting stats part of it comes in, which is that I want to modify this pair correlation function to take phenotype into account. So what we were doing before was just counting, is my macrophage and my blood vessel are units apart, and then normalizing according to the number of macrophages and the number of blood vessels, essentially. So what I do now is I say, well, instead of just saying, is this macrophage in my population or not, I'm going to have some target phenotype P, and I'm going to pick some kind of weighting function around that. So in this case, I'm thinking about P equals 0.5, and I'm just using a kind of triangular hat. So for every macrophage in my simulation, I'm going to look at it and say, what's its phenotype? If its P is 0.5, great. We're going to include that in our count. That's one macrophage. If its P is 0.4, maybe I say, well, I'm going to count that as like half a macrophage, because it's almost close to my kind of target, but it's not quite there. So I'm going to weight the contribution of every macrophage according to how close it is to my kind of target value P. And then I'm going to renormalize the pair correlation function according to that. And that ends up looking something like this, which basically has the same structure. So we're counting our cells if they're between these R units, but we're weighting it according to this W. And then when we do that, we just have to take into account, we're no longer dividing by the number of macrophages. We're kind of dividing by the total amount of macrophages that are matching this value. So to give you a kind of more concrete example of what that looks like, if I've got a load of kind of blood vessels sitting in a line at y equals 1, well, the macrophages here, these kind of dots, so this isn't really macrophages tumor cells, it's just synthetic data, but the dots are complete spatial randomness. So if I construct a pair correlation function from pink crosses to colored dots, it'll just come up flat and say there's no correlation going on here. But there is an interesting correlation, and that's in terms of what the label is associated with each macrophage, what its phenotype is. So in the left-hand one, that's just linear. They're just colored according to their distance from that line y equals 1. On the right, we've done a kind of quadratic. So practically, what you then want to do is calculate this kind of weighted pair correlation function. Well, with a normal PCF, you calculate for a number of different values of R. We want to do that, but also for a number of different values of P. So we end up with a kind of 2D metric that looks something like this. And the way you interpret this, so you have phenotype on the y-axis, radius on the x-axis, and basically any horizontal slice through this looks like a pair correlation function. So in particular, this is just horizontal slices of the graph on the right for various different target Ps. And so now you start to get a sense of where are points with label P relative to our blood vessels. So in particular, for the way we've set this up, we're expecting to see the strongest correlation on this line, P equals R, which is exactly what we do see. So this is the black dashed line. And kind of the width of that the width of each of these kind of PCFs, each of these peaks, is really determined by our choice of weighting function. So if we choose a very narrow target, we get a very tight line across that kind of predicted theoretical value. But obviously then we're bringing in potentially more noise because we have fewer points that match that criteria. Just to convince you that that's not a fluke, when we do this in the kind of quadratic case, we get this nice kind of peak of our PCFs matching this kind of nonlinear, P is R squared, which is exactly what we expect from this kind of synthetic data. So that's just to kind of persuade you that this does work. In terms of what then happens when I apply this to one of our kind of synthetic images, oh, that's blurry, we can interpret it in exactly the same way. But now our kind of y-axis is telling us how far along this kind of phenotype spectrum is our macrophage, and our x-axis is telling us what the distance is of that correlation or exclusion. So what you're seeing here in this example is at very short radii, my macrophages, which are very M2, are strongly clustered around blood vessels. So we're really specifically picking up these kind of perivascular niches now. We're saying if you look a very short distance away from a blood vessel, you're specifically seeing M2 macrophages. And in fact, you're specifically seeing the ones with a phenotype about 0.75 onwards. We're also getting this kind of population in the middle. So these kind of ones which are transitioning from yellow to blue. And we're able to pull out from here the distance that those macrophages are from the blood vessels. So in particular, we're seeing exclusion up to a length scale of about 15 kind of cell diameters, 15 units, which is kind of roughly the distance from the blood vessels to the center of the tumor. So we can apply this to all of our simulations from our parameter sweep, and you can kind of see that everything in the red box kind of matches that signature that I've just talked to you about. But you can apply this to whatever simulation you want, and it gives you a summary of kind of what's going on within there. You can also do, say, from tumor cells to macrophages. So, calculating this macrophage of phenotype P, where is that relative to the average tumor cell? And you can kind of pick up a difference, within, so that circle right up the top there is saying that this is really picking up that tumor cells have also made it to the blood vessels, right? Because we're seeing up there, M2 macrophage is very close to tumor cell, and from here is also very close to blood vessel. Whereas in the lower simulation, kind of don't know if you can see that red circle, but down there, we're no longer seeing the M2 macrophages with the tumor cells. So this is suggesting that in that simulation, we have a correlation at short length scales between these kind of transitioning macrophages and the tumor cells, but we're not seeing the tumor cells following the M2 macrophages back to the blood vessels. So yeah, if you want me to talk you through that in a bit more detail, I know this is a bit quick, but yeah, I can do that afterwards, no problem. So is this useful or not? Well, what I'm then going to do is say, well, look, we've just varied two parameters. Let's do a bit of a wider exploration of some parameter space and just generate some different kind of simulations, different realizations of what this looks like. And when I do that, I can kind of think of all of these statistics as describing a kind of fingerprint or a signature associated with kind of localization of cells. And it takes a bit of practice to kind of read what these things are saying, but these do describe where the cells are, which is quite nice. So it's quite difficult to then kind of look at how well these metrics correlate because they're not a scalar, which is a bit annoying. But what we can do is turn each of these different metrics into a scalar. So we can look at, for instance, the integrals of these WPCFs, or we can look at maximum values of the standard pair correlation functions, or we can look at the mean phenotype, and we can say, well, look, each of these statistics that I've talked to you about, you can derive from different kind of resolutions of data. So if we had statistics generated from some methodology, so PCFs from standard IHC, or if we know macrophage phenotypes from RNA-seq or something, if we can resolve phenotype or space, or both phenotype and space, do we get a better understanding of how all of these parameters are varying in my kind of big parameter sweep? And the answer is yes, and it gives you a massive table. So this is just some that I've kind of pulled out to make a couple of particular points. So if I'm just looking at cell counts, so these top two statistics are just the number of macrophages in a simulation and the number of tumor cells. Well, these correlate really nicely with this first parameter, P star, which is a macrophage extravasation rate. So you kind of expect that if I have higher extravasation, I can understand what's going on by looking at the number of macrophages. But you can see these other parameters. So these are kind of related to the sort of chemotaxis of tumor cells to macrophages and of the kind of rate of phenotype switching. are basically not correlated with cell counts at all. So if I'm just looking at cell counts, I can't really, I can't identify changes in parameters related to phenotype or changes in parameters related to my spatial location. If, on the other hand, I'm looking at statistics which describe our kind of phenotype distribution, so my average macrophage phenotype, or maybe the number of macrophages which have got an M2 phenotype, those are now correlating with parameters associated with phenotype, but I can't pick up changes in parameters associated with space, right? So hopefully the message of this is kind of becoming obvious, right? If I have parameters which are affecting either the localization of cells or changes in the phenotype of cells, I'm only going to pick up those changes in those things if I'm measuring those statistics. So if I have a system like the one I've talked to you about, where both space and phenotype are important, I'm only going to be able to understand that if I'm measuring from my images spatial statistics or phenotype statistics, or ideally both. And that's something that is kind of borne out through the whole massive table of this that I've kind of not really talked to you about. So, you know, there's a huge kind of demand for ways of analyzing, in particular, multiplex images, but also just imaging data in general. And I think this is an area where kind of as mathematicians, we can do a lot of stuff. You know, we have spatial statistics, we can adapt these things to take other variables into account. And so really that's where I'm putting my focus at the moment. But the other nice thing about these is, well, I've shown you these statistics applied to just agent-based models today. We can also apply these to imaging data. And that gives you a direct way of comparing your images with your simulations without just kind of saying, oh, well, we're seeing roughly the same thing if I look at this particular snippet. We can really kind of quantify the spatial interactions between these things. So yeah, I've brought the 7 bridge in as our bridge for this one. I thought it was thematic. But yeah, I think really, for me, this is where all of these things fit together. I think the modeling, the imaging, the spatial statistics are all kind of 3 sides of the same problem. And this is all up on bioRxiv as of last week. So yeah, it was a bit of a beast to write, but hopefully we've managed to get the story right. And I just want to say thanks to Helen, who yeah, all of this work has been with, and to all of these people for giving me money. Thanks very much.